We're going to turn our attention now to research for a little while, and I think this is the perfect opportunity to tell you a little bit more about what we do at NETRF. Our mission is to fund research to develop new treatments and discover cures for neuroendocrine cancer. And we have a unique space within the NET community. We're the largest global funder of NET research. We started in 2005, so we're not new on the scene. We've been around for a long time, but we have grown tremendously in the last few years. And we've invested more than $37 million in research. And I think as you hear some of the physicians talk about what we know and just as importantly, what we don't know yet about neuroendocrine cancer, you can see the need to keep advancing the science and keep funding research. So we know from an analysis that we did that neuroendocrine research, when you look at the prevalence of the disease, is underfunded by as much as 5 to 10-fold. And we've looked at our history and the number of grants that we've given and the people that we have funded. And we funded 2.5 times more NET researchers compared to the NIH and the National Institutes of Health and the National Cancer Institute. That's not in terms of dollars, but that's in terms of people, researchers who have entered this field and who we hope we have been able to maintain in this field. And make sure that they are able to look at neuroendocrine tumors, discover some of the intricacies of them, and help us develop more treatments. And we also fund around the world, so we're not just a U.S. organization. About half of what we fund in a given year is global, and we've really been able to increase that in the last 5 years or so. In terms of the results, we're very proud that the research that we have supported has resulted in 20 clinical research projects, including clinical trials where we test treatments, and you'll hear more about this in a little while. 8 patents that have either been granted or pending for new technologies or discoveries. And in basic and translational research, which is our sweet spot, that is a very significant achievement to have a patent on your work. 5 industry partnerships or spin-off companies have been created to bring treatments from the bench to the clinic. Having that sort of secondary investment in your treatment, the initial work has to be done, or we wouldn't be able to have those companies and have the interest from pharma and biotech in some of the work that we fund early on. 80 collaborations. We know that our researchers collaborate with their peers across the world, which is a great thing for all of you to know, because you don't necessarily see all of these scientists who are studying in labs across the world. But I think it's important and hopefully reassuring to know that this NET research community is working for each and every one of you on your behalf. They're dedicated to this, they are incredibly talented, and they're persistent, which is what you have to be in basic and translational science. And we also do a lot of work to fund early career researchers, because if they can't find early support, then they will probably go to another area of oncology or something else that they can study and know that they have some consistent funding and that they really can build a career. So that's an incredibly important part of our mission. And we're very happy that when we looked over the last 5 or 6 years, 100% of our early career grantees remain in research, 90% of them in NET research. And we're often the only source of funding for scientists who want to study novel, new approaches to treatment, kind of high risk, high reward types of scientific projects. And you can see, this is a little complicated, but you can see that discovery and that preclinical work, which is really a prerequisite to new treatments and clinical trials, that is, the 2 to 12 years that happens in the laboratories that really is absolutely necessary for clinical trials and for the process of bringing treatments to the clinic, to patients. And a great case in point for us is CAR T, which you may have heard of. CAR T is a type of immunology treatment, and it was first discovered and approved actually for blood cancer. And that happened at Penn. You may have heard the name Carl June. It was interesting because when this happened, one of the questions that came to our scientific advisors in our foundation, long before my time, but I think they were very visionary to go to University of Pennsylvania, seek out Carl June, and really connect him with someone who is in a laboratory down the hall, whose name is Xin Hua. And we made our first grant to Dr. Hua for CAR-T in 2014 and provided support along the way. So he was working in the laboratory, working on what a target might be for this particular treatment. So, you know, that's about 10 years of work and diligence. And probably in the next 2 months, a clinical trial will begin for CAR-T in neuroendocrine and some GI cancer patients. So going from laboratory work, can this be applied to neuroendocrine cancer, to a clinical trial in 10 years is actually fairly quick for science. And we're incredibly proud of that work and really the work that we do with all of our grantees. So, you know, we think it's important to make sure that the patient community knows about this. We know even now there are so many people who have innovative approaches and creative projects that they would like to do. We have probably millions more in applications than we have funding. So, you know, we're going to press on and fund as many of these people as we can. Any of you who would like to support us, we'd love to talk with you about that. And we actually have a success story and someone that I'm very pleased to introduce, Dr. Christopher Heaphy. He is from Boston University School of Medicine, and he's a cancer biologist who uses a combination of tissue-based, cell-based, and molecular approaches to conduct basic and translational research. Dr. Heaphy is the recipient of a 2022 NETRF Pilot Award for his work, and he's been working to profile the tumor microenvironment of pancreatic nets, and he's going to talk to us a little bit about that research and also what basic and translational research is composed of. So, Dr. Heaphy?
Well, thank you very much. It really is a pleasure to be able to present some of our work today and also talk to you about basic and translational research. So to start, I just want to give a little bit of background on basic and translational research, some of the terms. So basic research really are studies that aim to enhance our understanding of fundamental processes. So this could be biological, chemical, or physical. Sometimes you hear about this as bench work or preclinical research or fundamental research. Those are all similar words to describe this. It usually is motivated by a gap in knowledge, but I also really want to emphasize that this is the investment in the future of research, and this is what's building the ground. And from there, we can build up from there to be able to keep doing translational studies and definitely clinical studies as well. Translational research uses these basic science findings as a foundation for really translating discoveries into clinical application in medical advancements. So on the very bottom here, you have basic research, kind of basic questions of like, how does it work? Translational is how can it be applied to people? And then in clinical, how well does it work? What are the effects? And Dr. Ossie is going to be talking about clinical research and clinical trials next. So some examples of basic science research, maybe you've heard of these before. They could be in vitro studies. So this is conducted in a controlled lab environment. We're using cells or tissues potentially that are grown in culture, and then studying these where we can control the environment from how we treat it for a particular drug, how the cells are grown. All of these different ways, they definitely have limitations. There's advantages and disadvantages for each technique, but we use them in combination. And some of these models that you see on the right might be a zebrafish model, it might be a mouse model. There's definitely lots of different models that we could use. And then there's more and more that we're getting into in using computational models, so approaches to simulate biological systems, try to predict outcomes based on data that has been collected before, and try to analyze complex data sets. So we collaborate now with computational biologists all the time, and I think pulling this from different expertises is what's going to help make new discoveries as we go along the way. This is just an example of different assays or different models that have been developed and have definitely been used in the neuroendocrine tumor space. You can use tumor spheroids. So a lot of times cells are grown on a tissue plate, and that's just one cell line or one type of cell grown at one time. But as you can imagine, obviously cancer cells or other normal cells in tissue, it's not just one cell type, and it's not just grown on a flat dish. So there's obviously three-dimensional structures. So we're developing better ways to be able to model this and be able to mimic this. You might be able to get potentially a tumor specimen in which a patient has consented for research purposes, and then study that in three-dimensional context as well. And then you can use it for treatment purposes, trying to see what type of treatment may or may not work for a particular tumor. There's other assays that are being developed in terms of trying to mimic or model how we look at metastases. So you can obviously, all of these are extremely complex physiological processes, and we're trying to model it down to the single cell level and then be able to build up from there. So just to give some examples on some of the possible models that are being used. I also just wanted to give some representative examples where a basic science discovery eventually led to the development of a new NET treatment. So this has been in the past. And just to kind of give you a sense of this, there's lots of examples out there, but I just want to present a couple of them. So one is really the fundamental discovery that angiogenesis plays a role in cancer. So angiogenesis is the formation of new blood vessels. This is a completely normal process. It's definitely involved in development. It's involved in wound healing. But it's also now known that it supports tumor growth. So the person that really headed this entire field was Judah Folkman back in the 1970s. And a lot of those discoveries were based on other discoveries going back all the way to the 1930s. So you can tell that all these basic science discoveries, again, build upon each other and can make and potentially lead to new treatments. So now we have development of angiogenesis inhibitors as treatments for neuroendocrine tumors and other cancers as well. We've heard a lot about PRRT and other therapies, somatostatin analogs, but some of the basic fundamental discoveries originally was the identification of somatostatin receptor 2, or SSTR, that it's overexpressed in a lot of neuroendocrine tumors. So from there, we were able to build and start looking at all the different mechanisms. And what you can see here, I know this is a very complicated slide, but I put it up there because as scientists, we need to be able to look at each different pathway, see which molecules are being expressed and how they lead in those different pathways. And then we need to figure out that just in the normal pathway. Then we also need to look at it in a disease, a pathway in a cancer. And then if we start adding in treatments, how are we affecting those pathways? So these are the types of things and the controlled studies that we need to look at every time there's a new treatment that we're trying to evaluate. So of course, this has led to a huge, and you've heard a lot about it today, but huge areas in diagnostics and huge areas in therapy, and these are continuing on with numerous studies that are ongoing in the different areas that were outlined earlier today. I also just want to give you a brief overview of some of the ongoing quests that we're doing in the lab to translate basic science discoveries into potentially helping people with pancreatic neuroendocrine tumors. This, again, is just as an example of what we're doing in collaboration with a lot of other different labs and in collaboration with clinicians as well. So my lab studies telomeres. So telomeres are located at the ends of chromosomes. So you can see here we have different assays to be able to label the telomeres. So the blue is labeling the different chromosomes. The red are specifically labeling the telomeres. The telomeres are located at the ends of the chromosomes, and it's for the nucleotides, it's a TTA-GGG repeat. This repeat is repeated 1,000 to 2,000 times at every single end of the human chromosome. The telomeres themselves, though, really function to protect the ends of the chromosomes. So you can imagine that, take your shoelace, and when we used to have shoelaces with these little caps, at the very end, that cap is equivalent to what the telomere is doing. So it's located at the end, but it's also functioning so that your shoelace doesn't unwind, it doesn't tangle up with another shoelace. So you can think of that as those shoelaces as your chromosomes. And so really the function of the telomere is to prevent genomic or chromosomal instability. And so that's what happens normally. However, what can happen is that you lose that cap and you lose that telomere. So each time a cell divides, we lose a little bit of telomere DNA, 40 to 50 base pairs. And it really is actually, this is a normal process, it's acting as the cellular clock. So each time a cell divides, it loses a little bit more, and it is able to maintain and monitor how many cell divisions. And at a certain stage, in a normal setting, what would happen is the cell would undergo senescence, and it would stop dividing. And so that is actually a good thing. And you can see it here. So again, in the upper left, you see a chromosome. In this case, the yellow are the telomeres. This TTA, GGG repeat. The telomeres get shorter and shorter at a certain number of population doublings or cell divisions. In a normal setting, normal cell, it would stop. However, in an abnormal setting, especially in a precancerous cell or a cell that might go on to become a neuroendocrine tumor, this process keeps going on. Now you don't have those caps anymore at the end of your shoelaces. You can start to get a lot of recombination and chromosomal instability that occur, and this is what's driving a lot of the tumorigenic process. At that point, the telomeres get extremely short, but for a cancer cell to keep replicating and achieve cellular immortality, as you might say, then there's two different processes that occur, and they need to be able, that cancer cell needs to upregulate and maintain their telomeres. So this could happen through two different ways. One is telomerase, is an enzyme that gets upregulated. The other one is called alternative lengthening or telomeres, or ALT. So the first one, telomerase, it's present in about 85% of all cancers. It's present in stem cell. It does have a normal function. And I just wanted to highlight this because for telomerase, it was first identified in 1985, this enzyme, in a unicellular eukaryote called tetrahymena. And in the middle there's Carol Greider. She was a 23-year-old graduate student that first made the discovery in Liz Blackboard's lab. And they made it in this tetrahymena model system. It eventually goes on and is identified in all different cell types. It's identified in human cancers. And in 2009, they won the Nobel Prize for this discovery that has major clinical implications. So that's one pathway. The other pathway is this minor pathway. It's present in about 15% of all cancers, and it's this ALT pathway. And this is the pathway that my lab studies intensely right now. And we really got excited about this, or involved in this, a long time ago, and it was on a study that was done at Johns Hopkins, in which it was the first time pancreatic neuroendocrine tumors were ever sequenced. So they were looking at mutations in pancreatic neuroendocrine tumors. It was led by Nick Papadopoulos and Bert Vogelstein, and it was also funded by what is NETRF right now. And in this study, what they found, if you look at the spectrum of mutations in the pancreatic neuroendocrine tumors, they're very different than what you would see in a pancreatic adenocarcinoma. So pancreatic adenocarcinomas are the other tumor type, major tumor type that arises in the pancreas. So same organ, different tumors, different mutational spectrums, and you can see here, just comparing these two on these particular genes, they're very different. What really caught our eye was that this was also the first time that two genes mutated, ATRX and DAX, were ever found in human cancer. And it was interesting that in the pancreatic neuroendocrine tumors, this is pretty high. This is 43%. It was also, they were mutually exclusive, so you had a mutation in ATRX or a mutation in DAX. In that case, you never had them together. It really suggests that there's a biological function for a complex. So there was another group that was looking at this, and they said they thought they had some data to suggest that it also played a role at telomeres. So we were telomere biologists. We wanted to be able to collaborate with this with Hopkins group and be able to take a look at the telomeres. And what we found is that for the first time is, as you can see here on the right side, that we found some ALT-positive cases, and all the ALT-positive cases had ATRX or DAX mutations. So we knew ALT was a pathway that was there before, but we didn't know the genetic alterations that caused it. And this genetic alterations that cause it, when we do a telomere-specific FISH assay in a tissue, you can see these bright red dots. And those bright red dots actually represent telomeres that have been elongated abnormally. Since then, we've gone on to publish numerous studies collaborating with different groups that the presence of HALT associates with a poor prognosis in patients with primary pancreatic neuroendocrine tumors. If ALT is present, it's associated with larger tumor size, higher grade, advanced stage, metastatic disease. So one of the things that's really inhibited us in our pursuit of trying to translate these discoveries into the clinic has been the assay that we were using. So we have here two tumors, an ALT positive and an ALT negative case. We use a fluorescent-based assay that's really good in a research lab. So I do this in my lab all the time. In a clinical setting, it's really difficult to do. So being able to implement it has been difficult. More recently, we've collaborated with Atul Singhi at the University of Pittsburgh to develop a different assay, a chromogenic assay, that now we can be able to detect ALT positivity down in the lower right versus ALT negativity in the upper right panel there. So now we think we have an assay that can be used in a clinical setting. What's also interesting is that pancreatic neuroendocrine tumors, if I said, remember, 15% of all cancers utilize this ALT pathway. Pancreatic neuroendocrine tumors, about 31% are ALT positive in the primary setting. That goes up to about 75% in the metastatic setting. But pancreatic neuroendocrine tumors are not the only ones that utilize this pathway. There's other tumor types like sarcomas, which are found in soft tissues, like leiomyosarcoma, osteosarcoma at 60 and 64%. There's other tumors in the brain tumors that are also have a high prevalence for ALT. And then even a subset of liver cancer, this chromophobe hepatocellular carcinoma. So again, a discovery that was first made in pancreatic neuroendocrine tumors also has much more broader applicability to other tumor types as well. Things that we're working on in the lab right now are at ALT-specific therapeutic targets. So we do think this ALT pathway is cancer-specific, and so we're actually going and trying to target that specifically. So we have a couple different ways that we're trying to do it, looking at either the break-induced repair pathway or looking at synthetic lethality approaches. And looking at this in the cancer and also in the tumor microenvironment, we have grants through NETRF, the National Cancer Institute, and also the Department of Defense has a rare cancers research program that is funding part of our work. And these are areas that we're actively pursuing, and we're really excited about that. So I just want to kind of highlight in terms of basic science that, it's just two discoveries. The ATRX mutations are frequent in pancreatic neuroendocrine tumors, that these two mutations are linked to ALT-positive tumors, has now led to, that we also know that the presence of ALT is associated with a poor prognosis in pancreatic neuroendocrine tumors. We've developed an ALT assay that can be readily performed in a clinical lab using FDA-approved devices, and we do think this could be incorporated into future clinical trials. And right now in the lab, what we're really looking at is comprehensively understanding all the different molecular pathways and the best way that we can identify these ALT-specific vulnerabilities for therapies, specifically in NETs. Lastly, I just wanted to put this one slide up. So the AACR is the American Association of Cancer Research. It's a big association for cancer researchers in general. They had their 2024 annual meeting where 20,000 cancer researchers got together, and these were some of the future directions they were talking about in basic science. So again, this is for cancer research, but I think it definitely applies to NET research as well. So one of them is obviously embrace the complexity. These are complex systems. Systemic effects influence the cancer definitely, and the cancer has systemic effects. So we need to keep trying and determine what the best models are to try to look at these interactions. I think basic science discoveries are identifying new types of drugs that we never thought would be out there before, and I think be able to bring them in from different cancer types into the neuroendocrine space will be very interesting as well. And then multidimensional understanding of cancer. So we look at a lot of things in 2D or 3D, but I think we can also look at it in 4D if you think about it over time, so temporally over time, looking at different multi-omic approaches. As I said, I think computational biology is powerful and is going to help in these fields, and so we do collaborate with computational biologists. And I also think that just with everything else in our lives, I think AI is starting to come, or is here already. So in the research setting, AI is definitely helpful. I think at this point, it still needs human input, which I think is a good thing. But I think these two areas are really going to make huge advances in the field. So with that, I would like to thank all of you. It really is a great pleasure for me and privilege to be able to talk a little bit about basic and research in translational science with you. Also just want to highlight members of my lab, but also that the people that are doing these types of studies are PhD researchers, medical students, undergraduate students, graduate students, and of course all of this is in conjunction with our clinical colleagues. And we wouldn't be able to do any of this research without the support of all the different funding agencies. So with that, thank you so much.